Hello everyone and welcome to my video. My name is Mahdi.Far. Today and in this video we will learn how to use lithography technique for printing the molecule on the surface and doing click reaction and also protein immobilization. At the end, if you like my video, please push the like, press the subscribe button and also share it with your friends. Now let's start the video. For doing lithography and printing the molecule on the surface, we need to use this chip. This chip is called microchannel cantilever. It's a very small chip, maybe 5 by 5 millimeter. And also at the end of this chip, there is a channel. This channel is very, very small and you cannot see it with, I mean, naked eyes. And width of this channel is near 10 micron. That's why we call it microchannel cantilever. I mean, first you need to inject the ink and ink will go through the channel to the surface. Channel can transfer the ink to the surface, and you can print the molecule on the surface. For doing lithography, we need to use this machine. We need to mount this chip to this machine. This machine is called NLP. Here and in this video, you can see printing the molecule on the surface. You can see ink is transferring to the surface through this channel. Width of channel is 10 micron. It means diameter for every droplet, for every dot is 10 micron. Distance between two dots is 50 micron. But question is here, why should we use the lithography technique for printing the molecule? Lithography has some advantages. For example, you can print different molecules such as phospholipid, lipid, protein, and everything else. And with this technique, you can have very, very small droplets, in very, very small volumes, near femtoliter, 10 to the minus 18. And after printing the molecule on the surface, you can have a micro array. But here in this study, as I already mentioned in this video, we would like to use lithography for printing the molecule on the surface and doing click reaction and also protein immobilization. First, I would like to start with a click reaction. We would like to print the molecule to the surface and do two different types of click reaction. Just an example, I have selected SPAC and Thiol-yne coupling. SPAC is estrogen promoted alkylazide cycloaddition, and and this one is Thiol-yne coupling. First, you need to cover the surface with alkyne. First you need to functionalize the surface with alkyne. You have alkyne on the surface. In next step, for doing click reaction, SPAC, it's a click reaction between alkyne, cycloalkyne, and azide. In next step, we use lithography technique. We injected tamroazide. Tamroazide is a fluorescent ink. It's red. We injected tamroazide and we printed tamroazide on this surface. This surface contains of cycloalkyne. And we need to leave it for 1 hour. And after 1 hour, we need to wash it. With water, we wash the surface. And now you need to take a photo with fluorescent microscope and we will have something like this. You can see the red dots on the surface. Red dots, these are, tamroazide. When we have tamroazide on the surface, it means click reaction between azide and alkyne has taken place. And click reaction was successful. And here you can also see a scale bar. A scale bar is 50 micron. Distance between two dots is 50 micron. And diameter for every dot is 10 micron. The second type of click reaction is, again, when you have alkyne on the surface, you can do click reaction. You can do Thiol-yne coupling. It's a reaction between thiol and alkyne. Here in this study, we use the Cy5 peg thiol. Cy5 peg thiol, it's a fluorescent ink, and we injected the Cy5 peg thiol to microchannel cantilever, and after printing on the surface, we left the sample for half an hour, and after half an hour, we washed the surface and we took a photo with fluorescent microscope. Cy5 is violet, and again, you can see the violet dots. When we have violet dots, it means we have Cy5 peg thiol on the surface, and when we have Cy5 peg thiol on the surface, it means click reaction between thiol and alkyne has taken place. Click reaction was successful. If you would like to know more about click reaction and also surface functionalization, I ask you to watch the videos that I already published, and you can also find the link for them at the end of this video. We use lithography for printing the molecule and doing click reaction, and here we would like to do lithography for immobilization of protein. Here we have selected streptavidin. I mean, we would like to immobilize streptavidin on the surface with the help of lithography and printing the molecule. Again, we use the SPAC click reaction. We have alkyne on the surface. I mean, we have cycloalkyne on the surface. And here we use biotin peg azide. Here you can see a structure of biotin peg azide. Biotin peg azide is not fluorescent. And again, we injected a biotin peg azide to this chip here, and after printing the biotin azide on the surface, we left the surface for 1 hour, and after that we washed the sample. I mean, we washed the surface. And after washing, we covered the surface with FITC streptavidin. It's a type of fluorescent streptavidin. We covered the surface with this streptavidin, and we left the surface in a dark room, in a dark place with room temperature. And after 1 hour, we washed the surface. And after washing, again, we took a photo with fluorescent microscope. And here you can see the photo. You can see the green dots. Green dots are streptavidin. When we have streptavidin on the surface, it means we have biotin on the surface, because biotin has affinity with streptavidin. And every streptavidin can be connected to 4 biotins. I mean, every streptavidin has 4 binding sites for connection to biotin. And here you can see the green dots. When we have green dots, it means we have streptavidin on the surface. And when we have streptavidin on the surface, it means we have biotin. And when we have biotin, it means click reaction has taken place between biotin azide and cycloalkyne. You can see immobilization of streptavidin with lithography technique. So, hope you found this video useful, and if you like my video, please push the like, press the subscribe button, and share it with your friends. And also, if you would like to get notification whenever I publish a new video, please hit the bell. In this case, you will get notification. And if you have any question or comments, please write them in comment section. At the end, please don't forget that life is short and enjoy it. Thank you so much, and bye.